Hello, my name is Adam Metcalf, and I'm currently a senior scientist in the Aerosol Sciences Laboratory for Respiratory Drug Delivery at Philips. I'm here to present our work titled In Vitro Investigation into Fugitive Aerosol from a Novel Fourth-Generation Adaptive Aerosol Delivery System. Fugitive emissions are typically defined as aerosol that has been emitted from the patient or nebulizer and released to the environment rather than being directly inhaled and absorbed by the patient. As this was an in vitro study, we were only considering fugitive emissions from the nebulizer. Fugitive emissions have recently gained attention due to concerns that they may aid the transmission of COVID-19 and other viruses, but are also of concern due to caregivers or other people who are in the local environment of a patient where they may be unintentionally exposed to aerosolized drug. Fugitive emissions are particularly noticeable from jet nebulizers and constant flow mesh nebulizers that continuously generate aerosol during the periods between breaths and during the exhalation phase. This aerosol is typically emitted to the environment. Adaptive aerosol delivery, referred to as AAD, is a breath-activated nebulization technology designed to deliver reproducible doses of aerosol. The AAD algorithm monitors the patient's breathing and times pulses of aerosol into the inhalation phase of the breathing cycle for optimum delivery of aerosol with very little wasted as aerosol into the environment. The novel INEB Advanced Nebulizer has the fourth generation of AAD technology designed for faster delivery of aerosol compared to earlier generations. The INEB Advanced consists of three parts: the handset, the medication chamber, and the mouthpiece. For the experimental setup, three production-equivalent investigational INEB Advanced nebulizers fitted with non-metering chambers were washed and dried before use. Each nebulizer was weighed, then 1.7 mL of 2 mg per ml salbutamol drug solution was transferred to the medication chamber using a pipette. The filled nebulizer was weighed and connected to a filter attached to a breathing simulator, and the connection with the mouthpiece was sealed with parafilm. A second filter was placed near the exhalation port of the INEB Advanced mouthpiece to capture the fugitive emissions. The filter was set with an extraction flow of 60 litres per minute at a distance of approximately 1 centimetre directly behind the exhalation port, so that any fugitive emissions would be captured, but that the operation of the exhalation valve in the mouthpiece would not be affected. The nebulizer, breathing simulator and a timer were started, with the breathing simulator set to reproduce a USP adult breathing pattern. After the INEB Advance had indicated end of treatment and automatically switched itself off, the breathing simulator was switched off 5 seconds later. The nebulizer was weighed, and the two filters, one for the delivered dose and one for the fugitive emissions, were eluted, and the salbutamol content was assayed by HPLC. This process was performed in triplicate for each of the three INEB Advanced nebulizers. When we looked at the results, all of the delivered doses were very close to where we expected the values to be based on previous testing, and the intra-device coefficients of variation, that is, the variation in delivered dose for each device performing repeat treatments, were all below 3%. This demonstrated that each of the three devices was able to repeatedly deliver a consistent dose. The inter-device coefficient of variation, that is, the variability in delivered dose between the three devices, was 0.8%, which indicates that variability in delivered doses between devices was very low. The graph on screen now shows the results for the fugitive emissions from the INEB Advance. It shows that all 9 of the results for fugitive emissions were less than 0.3% of the mass of drug loaded into the nebulizer. Of the 9 results, 6 results were below 0.18% of the fill volume, which corresponds to the lower limit of quantitation of the HPLC assay, so these could not be accurately quantitated. These results are indicated on the graph by the number in brackets above each nebulizer. The primary advantage of AAD nebulizer systems has been the ability to deliver precise, reproducible doses of aerosol into different patient breathing patterns. The results from this study show that when tested into an industry-standard breathing pattern, the INEB Advance with the fourth-generation AAD algorithm produced delivered doses with low variability both from repeat treatments with the same device and also between devices. Recent publications have highlighted the advantage of breath-activated mesh nebulizers for the minimization of fugitive aerosol emissions, and in this study, the fugitive aerosol emissions from the iNeb Advance were all less than 0.3% of the volume of drug placed in the nebulizer. Thank you for your time listening to this presentation.